everyone! It's time to talk about molecular biology.
Today I'm going to talk to you about an interesting and important discovery in molecular biology, which is the CRISPR-Cas system. In this video, you'll discover what it is, where it comes from, and how it can possibly
change the future of mankind.
CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats.
Too many unclear words here.
CRISPR is originally a bacterial defense system against bacteriophages that are basically those little invaders that are just trying to ruin the lives of bacteria. Yes, the world of bacteria is cruel, and surviving it is really a challenge. Here's a little portrait of CRISPR. First, there is a leader sequence. Its role is to tell where to start reading the molecular code of CRISPR. Then there are repeats. Repeats are these identical sequences of beads, but what's really important is what comes between them. See those different sequences that are between repeats? Those sequences are called spacers, and they play a key role in bacterial defense. Those different spacers are like books on the bookshelf, each containing its own story. In bacteria, spacers are like books that tell stories of different bacterial diseases, and repeats are like bookshelves that keep those books to read them when they are needed. However, it is not enough to just store books. One needs to read them too. Because books of the CRISPR system cannot function all on their own, they need some helpers. Those helpers are Cas or CRISPR-associated genes. Cas genes contain the code to make molecular scissors, called nucleases, and molecular zippers, called helicases. Those nucleases and helicases, the Cas protein, combined with the CRISPR RNA, basically form
A dangerous foreign invader fighting team. Oh wait, the border is wrong.
So now the Cas protein, when the sequence is detected, cuts it up and thus destroys it.
The molecular war has officially ended without even having to start. See how smart those little bacteria are? Yes, I'm just one cell and still smarter than you, human.
But that's not all. Even though CRISPR-Cas is originally a bacterial defense system, it can also be used to defend us. This system can be used to edit human genomes and delete unwanted genes. Here's how it works. It's basically the same CRISPR-Cas system, but for this, we will need a specific kind of Cas protein called Cas9. The Cas9 protein combines with what is called the guide RNA, which would tell Cas9 exactly in which part of the genome to cut. Then, these two are going to be running around the genome until they find just the right matching spot. And then cut, and the unwanted sequence is out. Simple, right? At this point, the cell will recognize that there is some broken DNA and will start repairing it. At this point, we can insert new genes that will get inserted instead of the ones that were cut out by CRISPR-Cas9. This system can almost revolutionize genetic engineering and thus
change the future of mankind.